Okay, let's make sure everyone can hear me okay. Can everyone in the back hear okay? Good? All right. Well, thank you so much for the invitation to be here. I always welcome the opportunity to talk about my research. I'm very excited about it, and I hope I can interest you in it too. So, while I'm technically on vacation, actually what I've been doing was I was in New York City trying to find an apartment because we move in a few weeks, and we actually found one, so we're happy about that. So, as Trudy mentioned, I've been studying Parkinson's disease for over 2 decades, and I was really intrigued by this because of the complexity of the disorder. So we know that increasing age is the biggest risk factor for the disease, but there's also been data that, for example, exposure to different pesticides can increase the risk. There's certain genes that increase the risk of getting Parkinson's disease, as does head trauma as a risk factor. And then there's 3 very interesting factors that actually decrease the incidence. So caffeine consumption decreases the incidence, so drink your coffee and tea. Exercise decreases the incidence, so be active. But cigarette smoking also appears to decrease the incidence, and I cannot encourage that activity. It's known to increase the risk of like 200 other disorders, but it's still a very interesting fact because it's some sort of environmental contributor that's changing the risk for the disease. And so in Parkinson's disease, it's really characterized by the black substance in the brain there. It's called the substantia nigra, for black substance. And in Parkinson's disease, those neurons die, and that black substance goes away. And so one of the big challenges has been what makes those cells in the brain die faster than the other cells in the brain? And so this has been a big interest of my research. And it wasn't just from a scientific part, there was kind of a family link to this. So from a family history standpoint, my grandfather, Paul Miller, worked for the Department of Agriculture. He was a plant pathologist, studied plant disease. He was also a good friend and colleague of George Washington Carver. And in Carver's biography, they actually cite my grandfather's work and their collaboration together. And then, as you know, my father, Dick Miller, also worked for USDA. And I remember as a kid going to work with him and seeing these cows with these earrings on. And the earrings contained pesticides. And the idea was to keep the flies and things away from the cows. But it was kind of seeing that balance is that pesticides can be very useful for controlling insects that spread diseases, but it still can be a concern for other conditions. The picture there is my father and my oldest son graduated from Emory a couple years ago. He's actually not here, but my other kids are in the audience. And so this has been something that, having a grandfather had the disease, and having a family who's worked in agriculture and had exposure to pesticides, this was always a very big interest. But in my training in toxicology and studying chemicals, I was always kind of bothered by when people would say, well, pesticides are a risk factor. Because when you think about pesticides, you have all these different classes of chemicals. You want to kill rats, you want to kill fungus or certain plants. You often want to kill insects, and there's all these different ways of doing this. And the chemicals are so different. They could be as different as any sort of set. You have different structures, different types, and they're used different ways. So looking at pesticides as a group doesn't really help very much. All we can really do as scientists are study individual compounds one at a time. And this was something that I found as a researcher 25 years ago, was we were trying to look at one chemical at a time, and that's not how we're exposed to things. And so when you think about what sort of chemicals could be involved, you have to kind of go back historically, because if you're studying a population of people that have Parkinson's, say 20 years ago, they were born 80 plus years ago. And so what were the chemicals that were introduced? So for example, something like DDT wasn't introduced until the 1990s, but that's something that you could have an exposure as a child or something could have an impact. And even things like we phased out DDT in 1974, but we can detect the metabolites of DDT in almost everybody because it's a very persistent chemical. It stays in the soil, it stays in the plants, it stays in our bodies for a very long time. And so a lot of this is looking at what sort of chemicals have been introduced, but really the only thing you can rule out is that if you were studying people in 1990, the newer neonicotinoids that people are concerned about with bees probably had no role because it's a relatively new insecticide. But all these other ones, even that we've banned and phased out, they still persist in the environment. So there's still things we're concerned about. And so what we do in the laboratory is we take we culture different types of cells from brains, and we study individual cells in petri dishes, and we can treat them with individual chemicals, like this, known as a pesticide that organic gardeners like to use because it's a natural pesticide, but it's also a really good poison for cells. And so even though it's natural, it doesn't mean it's safe. The idea is that synthetic chemicals are designed to do something, but nature's also very good at doing things that you may not want to have happen. And so we can study these cells. My lab has been really interested in studying animal models of disease. And one of the most interesting examples we've had is we found that one of the really important parts of the brain is that we know that in Parkinson's disease, there's a lack of dopamine in the brain. And so the main treatment is to give the precursor to dopamine called L-DOPA. It's what's in Cinemet. And so you put the precursor of dopamine back in, you make the dopamine, and it helps with some of the symptoms. And so what we did, we found that animals that couldn't store dopamine well would develop a Parkinsonian phenotype. And so we took animals and using transgenic genetic technology, reduced their ability to store dopamine in the brain. And after a couple years, these mice developed a Parkinson's condition. And just to show some examples, first is that their dopamine levels are reduced about 90%, same thing you see in humans. If you look at their ability to walk on the far right, you see that the stride length of those mice is shorter, and it resembles that sort of staggered, slowed gait you see in Parkinson's. But this slide here shows that if you take these mice that can't walk as well, and you give them L-DOPA, their stride length goes back to normal. So they have the same response to drugs that humans do. So it's a really nice animal model to study the disease. And when we compare other factors with this, is that we call these our VMAT low mice. VMAT is the name of the protein that we knock down in the mice. It has the same depletion of dopamine. That black substance, those neurons die, just as we saw in humans. There's also a loss in norepinephrine, which is another neurotransmitter that actually comes next to dopamine in the pathway. And this was interesting because that work where we showed that heart levels of norepinephrine were depleted was done in collaboration with a colleague of mine, Dave Goldstein, at NIH, right down the road from here. And I've collaborated with him for several years on this. So we have these mice that have all these very similar comparisons, but the idea is that we disrupted the ability of the mice to store dopamine in their brain. And what evidence is there that something like this happens in humans? And so this was something that over the time we were studying our mice, several papers came out where they showed mutations in that same gene caused a Parkinson's-like condition. So it caused it in very young children. These two papers showed that people that overexpressed this VMAT2 protein actually had a decreased incidence of the disease, which kind of made sense. The idea of we knocked it out, it was bad. If you had more of it, it was better. And then just a couple years ago, this paper came out on the right where they suggested that this dopamine storage was one of the problems. And I was really struck by this because I recognized this name Ola Ornikevich because in the field of Parkinson's, he was very well known because in 1960, well before when I was born, he just reported that dopamine was low in Parkinson's brain. So this was always, this is the first thing you learn about Parkinson's disease in school, and Ola was the one that discovered that. And I started doing the math, and I'm like, how old is Ola? He was publishing that in 1960, and he was also the first to show that L-DOPA worked in it. And it turned out that he's still an active scientist. I was at a meeting last year in Austria, in Vienna, and I got to meet Ola Ornikevich, and it was on his 90th birthday party. And I saw just last year he published another paper. So he published at 91. My wife's asked me when I think I'll retire. I have a new goal. 91 seems pretty good. And so he's still doing really good work there. It's really nice to see that. But it was nice because it showed that there were some very human connections to what we had seen in our mice. And so when we looked at this, what we had done, we'd shown that when this vesicular function, this ability to store the dopamine inside the cells, is down, those neurons, those dopamine neurons in the brain are more likely to degenerate and die in the mice. And so we'd done that with mice that we'd knocked out the gene and had low levels. And our question was that, what would happen if you went the other way? what if you could increase the ability to store dopamine in the brain? Would that be a good thing? And so we went back to our genetic technology and to see if we could make mice that overexpressed VMAT2, this protein that stored the dopamine inside the cells. And so we did this a few years ago, and it worked really well. That we made these mice that had increased levels of this protein, they could store dopamine better, their brains actually have more dopamine in them than a regular mouse. And when you stimulate these neurons, they release more dopamine, and so they actually have more action. And so this was a really exciting finding because it shows that this might be a way to improve the function of these dopamine neurons. And so this is something that I was in Europe a couple weeks ago, and I met with people in a drug company who are interested in targeting this protein for therapy because they think it might be a way of improving dopamine function in these neurons. And it was interesting, they contacted me, and it was very much based on the work we had done in this paper showing that this would work in mice. And what's really exciting, when I first was thinking about this, is that VMAT also packages norepinephrine and serotonin, was if you made mice that overexpress the storage and release of these things, that would it be a good thing? Like if you had too much dopamine and norepinephrine and serotonin, would the animals be manic? Would they have this sort of effect of having like amphetamine or a drug like that? But it turns out they're just really happy mice because they have a little bit more dopamine. But the idea is we can age these mice out till as old as they can be, and we see no adverse effects of having this improved storage of the dopamine. So this is something that got us really interested in how that storage of dopamine in the brain was really critical. And then a couple years ago, I saw this paper, and it was a group of geneticists that were studying Parkinson's disease, but they had data on people's smoking history. It's very easy for people to recall how much they smoked over their lifetime. And what they found was that they identified a gene that was associated with the ability of cigarette smoking to prevent Parkinson's. Remember, that was a very weird finding, that we know this, but it's still very interesting. So when I saw this, I'm like, well, again, we don't want to encourage people to smoke, but if we could figure out why it's doing it, maybe we could target that gene. And so it was interesting, they did this study, and they found the gene that showed this effect in humans, but they also used flies, like the fruit flies, Drosophila, that people have used in genetics for a long time, and did a model of Parkinson's in the flies and found the same related gene in the flies. And when I saw this, I was really excited for a few reasons. One, it wasn't just the smoking piece. There's been studies that have shown that nicotine-type compounds in foods also decrease the incidence of Parkinson's disease. So it's not just about the smoking. There is something in the nicotinic activation that could be protected to these neurons. What really grabbed my attention was the name of this protein. It's the synaptic vesicle glycoprotein 2C. It lives on the vesicle right next to VMAT, which my lab had been studying for 20 years. And so we decided to start studying this new gene, SV2C. And so one of the ways we do these things is that if you want to know what a certain gene or protein does in a mouse, you genetically delete it. So we make these mice that didn't have any SV2C. This is the protein in their brain. Then we knock it out, it's all gone. They can't store dopamine as well. It's not as dramatic as we've seen in the other mice. But we were able to show that this SV2C is controlling the ability to release dopamine. And so we think this is another one of these targets that might enhance this effect. And so, again, very little was known about this protein, very little research has been done on it. And so I asked one of my students to, we figured out a way to look at the protein in brains. And so I said, can you get some tissue from our brain bank at Emory, where they have people that have donated their brains, we have postmortem samples that had, control patients, Parkinson's, Alzheimer's. And we started looking at the expression of this protein. And so this is what it normally looks like in control brain. But in Parkinson's, we saw this really strange expression of this SV2C protein. And this pathology had never been described before. And so we reported this, and we also showed that we didn't see it in Lewy body disease, multiple system atrophy, Alzheimer's disease, progressive supranuclear palsy. It was only in the Parkinson's disease brain. And so we now, based on this work, we got a grant from the Michael J. Fox Foundation to look at the ability of manipulating this SV2C gene to see if it could be a therapeutic target for Parkinson's disease. And so this has been, when you kind of how we've been working in the field for a long time, is that we look at individual genes and individual exposures. But we know that our risks to certain diseases, even genetic ones, are due to a combination of genes, and they're due to a combination of exposures and a combination of those combinations. And on the genetic side, we're getting better at figuring out the multiple genes that are involved in certain diseases, but we're still lacking that ability on the environmental side. And this is something that had been troubling me for a long time. And if you think about the progress we've seen over the last 100 years on the genomic side, it's really been kind of the century of the gene. Going back from when NIH was founded, the structure of DNA by Watson and Crick, and this is not to disparage, I love genetics, I'm a big fan of genetics. They announced their discovery at the Eagle Pub in Cambridge, and there's me drinking a beer when I visited the pub a few years ago. So I'm really a big fan of genetics. And there's been a lot of great discoveries, and so the Human Genome Project, which was started in the late 1980s, was able to sequence the entire human genome. And one of the really interesting facts about this was that when the Human Genome Project was started, with the existing technologies, it was predicted to take 115 years to complete that project. But they knew if they put the money into it, the technology would advance, there'd be investment from companies, and sure enough, it was all done in about 10 years. And so again, the idea is that genomics was pushed forward because of this dedicated effort and money at a worldwide scale that went into going after that. And we've gone to this point now, so this was a $3 billion project, and just last year, Craig Venter, who was involved in that sort of race for the genome, his group had a paper published where in a single manuscript, they had the sequence of 10,000 people. So like the Human Genome Project was this huge project. Now Venter's group has a paper with 10,000 full genomes. It's just amazing. And then Beijing Genomics now can do a full genome sequencing for like $600. It's a China-based group that's been very good at this. How many people have done 23andMe or Ancestry.com? So you're among about 4 million other people that have already done that. You can get a lot of genetic information for just $100. So again, the idea, we have all this amazing genetic technology. And so, this sort of genome concept, the gene was the first ohm that was determined, but we can look at the RNA, which is the transcriptome, all the proteins is the proteome, and it's really become known as the compilation of all those contributing members of some biological domain. But it's also been kind of funny is that a lot of people have created their ohms for whatever they want to do. There's a website that lists all like 300 different ohms. My favorite is the ridiculome that someone coined for all the sort of over-naming these things. But when you think about it, it's when you think about how nature and nurture contribute to these different things, we have the nature side taken care of very well. But when you think about the nurture and environmental exposures, these things are not studied at that same scale. So again, you look at the DNA side, we can get whole genome sequencing and get tens of millions of features about someone's genetic information. But if you want to see what people were exposed to over their lifespan, we might get, from an environmental-wide study, maybe data on 400 different chemical exposures, even though we know that through industry we've made about 90,000 synthetic chemicals. You think about all the things we eat in our diet. Plants have a couple 100,000 chemicals that are plant chemicals we bring into our body, but we still only measure a few hundred of these things. So it's nowhere near the scale that we need to be. And so this is, I always felt like science could do better on this. And when you think about NIH, the paradigm has always been to identify a disease and then try to figure out that cause and come out for a treatment for that malady. It's the National Institute of Disease is really what it is, right? We don't think about the health standpoint. Very little of that money is spent on attaining optimal health, and almost all those things are non-genetic. Right? And the idea is that if your doctor wants you to improve something, they're not going to go, let's go change your genes. They make you modify your behavior. They make you change your diet. They make you get more exercise. These are all non-genetic things that we use for interventions. And so this got to this point where we felt that we had to have an approach where we could look at the environmental contributors at the same scale as we were doing for genetics. This has been the big goal. And so this is when this concept of the exposome came around. So Chris Wilde, who is the director of the International Agency for Research on Cancer at the World Health Organization, this is the group, when you hear that something's been named a carcinogen, this is the group that makes that official edict from World Health Organization. And so he defined the exposome as all exposures throughout the lifespan, including lifestyle, diet, and environment. So really the totality of all those exposures. This was something a friend of mine, Dean Jones, and I at Emory had also provided some of the refining of this definition of the exposome to also include how the body responds to it. So if you've heard of epigenetic changes, like when you have these changes to modifications of your genes, these are things that are typically reflections of past exposures. So if someone had smoked previous in their life, you can find epigenetic marks that'll stay there for 50 years. We think the same things happen with many other chemicals, is that so how your body responds to it can help identify those sort of fingerprints of past exposures for chemicals that may have gone by. So we really like this concept of the exposome, but we didn't really know how to do it. This was the challenge, like how do you actually measure something so complex as the exposome? And so we started working on this at Emory. In 2013, we had a grant awarded. There was about $5 million from NIH for our Hercules Exposome Research Center. And the idea was that we don't really know what to do, but we know we need to improve the tools. We have to get better at measuring these complex exposures. And so we had this was the first center grant in the United States funded on the exposome. It was just renewed for 5 years a few months ago. And it's really about kind of pushing the different technologies and promoting the concept of the exposome. And I'll get into some of the data. This humanexposomeproject.com website was something we developed for that. I am taking that with me to Columbia, so I'll be updating that over the next few months. I also wrote the first book on the exposome, came out a few years ago. I'm supposed to be writing the new version. I'm really way behind on it. But it was, again, this is something where there's no other book on the exposome. There was nothing out there. It's such a new field. And so we've really been kind of pushing this forward, and I'll explain more of how we've done this. So when you think about the century of the genome, when you get to the exposome, it's barely a decade of this. This has been very new, where Chris Wilde coined the term in 2005, and then there have been, the European Union has funded a few projects to kind of work on the exposome. Our center was funded. Other universities and schools have started investing more effort into this. And I think we're really starting to see this take off now. So multiple universities have held workshops or symposia on this, Yale, Harvard, Stanford. There's been articles in Wired, Popular Science, Scientific American. So there's a lot of interest in how we can scale up our evaluation of the environment. And so this is where, again, our research at Emory has been very focused on how to do this. And so to kind of give an example of some of these things, when you think about our ability to do chemistry, if you go back to 1957, we had systems that could measure all 20 amino acids that make proteins up in our body. And this was important because there's the diseases that have these amino acid problems. So we could measure 20 features at a time. 50 years later, we could do 200. Think about what happened on the genome in those 50 years. They went up orders of magnitude. We went up 1 order. Very little on that. But then just over the last 10 years, with new mass spectrometry technologies and separation techniques, we've gone from a couple thousand to 10,000 to 20,000 to the point we can now measure over a couple 100,000 features in a single drop of blood that represent all these things that are in our body, all these exposures, the drugs we take, the food we eat, the pesticides that are on those foods, air pollution sort of exposures. We can measure all these things in single runs of mass spectrometry now. And this has really been, transformative in our ability to get to that same scale that we've been doing with the genomic side. And just to give some examples of some ways we've applied this in some early studies, I'm not going to get deep into the data, I just want to illustrate some of these things. We did a study of people with Parkinson's disease, and so we had patients at Emory University and controls, and we took a drop of their blood, we ran this analysis, and we could detect all these different drug metabolites they were taking. We could detect different antioxidants in their diet and different parts of this, and we could very easily separate Parkinson's from control. In this study, what we did find was that much of what we were measuring was due to the drugs they were taking, and that makes sense because you're taking a strong drug that's affecting dopamine, it changes it. And so the next study we're working on now is with a group in Europe where they have about 600 patients with Parkinson's disease where they collected blood samples from them before they were diagnosed. So it was a very long, like 50-year study, but they have the blood samples before they were even diagnosed with the disease. And so that'll help us understand more about which of these factors might be predicting whether or not a person develops Parkinson's disease. And so we're able to do these things on very small samples. We also did a study on Alzheimer's disease and mild cognitive impairment, one of the precursors to Alzheimer's. And so this was in collaboration with people in neurology at Emory. And so in this study, we did a couple different versions of it, but we had about 60 control patients, about 90 Alzheimer's patients, and about 50 with mild cognitive impairment. And we applied this same sort of technology to them, and there's all these different things that we can measure in their blood. We see changes like in dietary factors, but one of the really striking things we saw was this metabolite of DDE, which is a metabolite of DDT, right? We banned it in 1974, we don't use it anymore, but it was enough. We still saw that in these patients, and the patients' average age were like 70 or so. And what was really interesting about this finding was that we were doing this in this unbiased way. We don't know what we're looking for, we're just looking for everything. And there's hundreds of thousands of choices, but a paper came out about 3 years ago that showed elevations in DDE in Alzheimer's patients in Texas, totally different population. And so our sort of unbiased and untargeted approach found some of the same things people had found when they were looking for pesticides. And so it was a really neat connection to show that this technology could pick up these things in this very high throughput way. And so one of the important things here, when you're trying to do studies of complex diseases, the cost is a factor. Because if it costs $5,000 to do analysis, you can't do it on 10,000 patients. There's just not enough grant money out there to do it. And so what's exciting about this is that if you want to measure all these different chemicals in humans, you have to run multiple assays, all these different technologies, all these different mass specs. It would definitely cost about $3,000 to do, probably $5,000. And we can do each of these 2 assays we do for $125. So for $250, we can give information on 100,000 sorts of features. So we're pretty much getting 10 times the information for a tenth of the cost. And so really kind of scale things up, a couple orders of magnitude. And so we think this is really going to help us move forward with the sort of large scale and precision medicine thing. So you probably saw information about the All of Us research program, which was officially launched a few weeks ago. This is NIH's big precision medicine initiative part, where they want to enroll 1 million patients into the study. And so the idea is to get a very diverse population of people, all different conditions and things, and you're going to have 1 million different people come in. They're going to have blood samples from them that will be biobanked at the Mayo Clinic, and you can apply to do different things and study these conditions. And so we think this is a really neat opportunity, and one of the things I was working on with my move to Columbia is I formed a consortium with Columbia, Harvard, Emory, and Brown to see if we can scale up this sort of exposome type measure to really work on the sort of precision medicine activity. And so this is exciting because Columbia is one of the leaders in this sort of precision medicine space. They've put a lot of money, the president's very much behind it. One of the first things they set up was the Laboratory of Personalized Genomic Medicine. This is what we do. We do the genes first because we know how to do that, and we wait later to figure out the environmental part. But the Irving Institute at Columbia that does a lot of the clinical and translational research, they said, we want to do this. We want to help build a core to do this exposomic, metabolomic work that my lab had done at Emory. And so I'm working with them to set this up, and hopefully in a few months after I get there, we'll be doing that. Another exciting thing about my move to Columbia, this YCAP study, led by Richard May, who's the chair of neurology at Columbia, has been studying almost 6,000 residents in the Washington Heights area they're about a third African American, a third Hispanic, a third Caucasian, and he's been following them for like 25 years. he was following them before they were diagnosed with Alzheimer's or mild cognitive impairment. So he has those blood samples before diagnosis, and he's been following them for decades, and we're writing a grant together now to do this analysis on them. And it was something we just couldn't do 10 years ago, and so it's really exciting to be in this position to start pursuing that. Just to give you an example, like what this sort of data looks like. So the Mayo Clinic contacted me about a year ago and said, we're studying this liver disease. It's called primary sclerosing cholangitis. The only treatment for it is a liver transplant, so it's a very severe disease. And they said, we've done all our genetic tests on them, but we can't figure out who's most likely to need the liver transplant, who's going to develop the disease. They really had no information. And so they said, can you do this sort of exposome analysis on the samples we have? And so they sent us a couple hundred of them. And what we're able to do is look at how certain chemicals in the blood are changing the chemicals that are part of our regular metabolism. And so it helps identify chemicals that might be contributing to it, or might just have a hard time processing because of the disease that's going on. But what was really exciting about this study was that we had shown them that we saw this altered bile acid metabolism, and bile acids come from the liver, and so I assumed that was just because they had the disease. And he said, and he told me it was really surprising because he said those patients, the samples we gave you, were clinically normal. Their blood work was normal in their normal labs, but our technology could pick up alterations in liver metabolism that they couldn't detect with the clinical chemistry. So the idea is these sorts of technologies might help monitoring progression of disease, but also help identify some of the chemicals that might be changing these different features. So we actually have a big grant with them that we think is going to get funded to study actually several hundred of these patients. And so I think this is what this type of work we can apply to the Precision Medicine Initiative and really scale up to tens of thousands of patients. So I was talking about my mice before and work with flies is that studying humans is the most relevant population for human disease, but we can't treat people with chemicals to see what they do. Like we have to use our animal models for that. But it's hard to do mixtures and complex genetics in mice. It's very expensive to do. And so what my lab has started doing, for many years in genetics, the nematode C. elegans is about a millimeter long worm. All the genome has been studied. It was actually one of the first full genome sequenced before the Human Genome Project. So we can study these little worms. We can grow them up by the hundreds. If you want to have a gene knocked out from them, you just order them from a catalog for $7. They've already been done. It's amazing. You go to the worm catalog and just order them. And so we can order worms that have genes mutated in Alzheimer's or Parkinson's or ALS. We can order all these different worms, and then we can expose them to many different chemicals at a time. We can expose them to 100 different chemicals at a time. And this technology we use, this exposomic mass spec stuff, works perfectly on the worms. And so we can measure all the chemicals and the metabolites of what we expose the worms to using the same techniques we're using in the humans. And so it gives us a really great way to look at gene by environment interactions at a scale you just can't do in laboratory rats or mice. It's just too expensive to do. And so the idea is we want to do the human studies, and then when we see these different alterations, we can go back in the laboratory and replicate them in the C. elegans nematode model to kind of test those hypotheses and see what's happening from that. So it's really kind of a nice way to go from that reductionist laboratory side up to the human population. Some other kind of fun things that we've developed. A friend of mine at Georgia Tech, Mark Prausnitz, you may have seen some of these in the news about these microneedle patches to deliver flu vaccine. So he developed these patches that it's like a little Band-Aid, and it has glass needles that dissolve in the skin. And so he put desiccated, dried out flu vaccine into them. The person wears it for a few hours, it dissolves into the skin and delivers a flu vaccine. It works very well. But if you're trying to deliver a vaccine to areas that don't have refrigeration, you don't need refrigeration. It's an amazing thing for developing these things out. And when I talked to him a few years ago, I'm like, could you make a patch that did the reverse, that actually took out the fluid under the skin, like it comes out from a blister? And he was like, he's an engineer. He's like, yeah, it's just an engineering problem. So we gave him some money, and he invented this. So he developed these patches, and what you see here, this circle is a hydrogel that is enlarging as it's absorbing that blister fluid under the skin. And he can get about 10 microliters, like this is like a fifth of a drop, and it's very small amounts. But we can extract the liquid from that, and run our same exposomic analysis on it. And we can detect thousands of chemicals in this tiny amount of interstitial fluid. And it doesn't involve someone going to the doctor for a blood draw, because they can just wear this patch for a few hours and then mail it back to us and do this analysis. So it's still in development, but I think it's going to really help us do these sorts of big studies on tens of thousands of people, because you can cut out that whole doctor's visit and phlebotomy. And people are now using some of these technologies to monitor blood glucose and things. We can detect many things that are normally in blood. But this has been a really kind of exciting development, because it was a technology that was really critical for us. So, we think what we're getting to now is that we want to assess how all these exposures, to industrial chemicals and pesticides and, the medications we take, all of these factors, how they affect our omes, the genome, the epigenome, the transcriptome, and the sort of the metabolic features. And it's important because these metabolic changes are the closest thing to what you see when you go to the doctor. When you go to the doctor and you have your blood pressure's increased or something, it's something you can detect in the metabolome. Your genome doesn't change every day, but your metabolome changes after you eat a meal or something. So it's very sensitive to those quick changes. And I think it's going to really help us understand this connection is by using these sort of omic technologies to connect the clinical side to what we're actually seeing from exposures. And so this has been a big drive of this. So just to kind of summarize here, what I've seen the last decade or so is that we have a very comprehensive view of the genes. We can do this whole genome sequencing, it's amazing. A very selective view of the environment, and this is why we have a genetic view of disease. Ask a geneticist what percentage of diseases are genetic, and for complex disease, let's say about 20%. The majority of causation of complex diseases are non-genetic, but we're so good at doing this, we have this very genetic view of disease. And I think what the exposome can do is help balance out this equation. If we get that comprehensive view of the environment, we'll start seeing a more balanced view of disease, which I think will be much more accurate. And so I think this is where we're getting to now, of having that level of features that's truly comparable to what we're doing with the human genome. So I just wanted to thank all the people in my lab that have been doing this work. Thank my parents for inviting me here. And if you want to hear more, I was interviewed by NPR a few weeks ago. If you Google Big Picture Science Exposome Blog, it'll take about a 15-minute interview where I talk about the exposome. It's a nice little summary there. And again, I'd be happy to answer questions immediately after and even after people depart. So with that, thank you very much, and I'm happy to take questions.